and I have the pleasure to introduce Dr. James Trosko for the next presentation. Dr. James Trosko is from the Michigan State University in the US, and among other achievements, Dr. Trosko has demonstrated UV-induced DNA damage and repair in normal human cells, has discovered a lack of DNA repair in syndromes of xeroderma pigmentosum and cognate syndrome as well, has demonstrated that tumor promoters inhibited gap-junctional intercellular communication, and has demonstrated that chemopreventive agents prevented the downregulation of gap-junction intercommunication by tumor promoters. Dr. Trosko will speak about understanding complex mechanisms in determining adverse and beneficial health effects with nutrition diets, from basic science of hazard identification to the concept of one health, one planet. Dr. Trosko, the floor is yours.
Thank you. I wish to acknowledge my deep appreciation to EFSRA, to the co-chairs here, to the fellow speakers, to my peers in the audience, ladies and gentlemen. I'm indeed flattered to have this opportunity because basically my peers know that I'm an iconoclast. For 50 years studying radiation, chemical, and biological toxicity, I've been challenging many of the major paradigms. Now, how do I follow the lecture that just preceded me? How do you follow a lecture on sex? Well, the only thing I can think of that now ties every one of our speakers, and especially the last one, to what he said, our purpose is to reproduce. I remind you of Theodosius Dobzhansky, the famous evolutionary geneticist. By the way, that's my previous training. He once said, "Nothing in biology makes sense except in the light of evolution." And today, talking about what do we need to know for proper, rigorous risk assessment related to diets and nutrition. And that's what I'm going to talk about. You see in the first slide, after I wrote that incredibly long title, I decided to have the next slide, which makes it more specific. Any one of you looking at these two diets who nutritionists and epidemiologists have pointed out are the two best diets on earth. How can you compare okonomiyaki to pizza? How can you compare mochi to a cannoli? How can you compare green tea to vino rosa, all right? So obviously there's something beyond appearances that have made these two diets in the minds of nutritionists as the best diets. Now mind you, today, if you look at why they decided that, you see the patterns of aging and diseases of the people in Japan are different than the patterns of aging and diseases in Sicily. I happen to work in both places, and I happen to like both of their diets, and I could afford both diets. Today, these diets are not affordable. To have bluefin tuna caught in the Mediterranean, shipped to Japan, cost about $50,000 per fish, all right? So only the Sony CEO can eat it. The point I want to make here is, beyond appearances, there must be molecular mechanisms in the composition of those foods that either enhance the risk or reduce the risk to cancer and other diseases. I'm not going to spend too much time because I want to get to some new data that I want to show you at the end. In effect, really, I'm going to tell you the end just in case they're going to shut me off, okay? I'm going to tell you what I'm going to tell you. I'm going to tell you, but I'm going to tell you at the end what I told you. But I'm going to tell you what I told you right now, all right? And that is the basic metabolic problem globally, both ecologically and human-wise, is a collision between the glacially slow genetic changes we need to make to generate energy through food for reproduction. And there's a tie between me and Dr. Sarp now. We all have to reproduce, and survival. And it turns out the biological basis, which took over millions of years for our pre-human ancestors to survive, collides today, especially within the last 50 years, with cultural evolution, bearing on our nutrition and diet. And we can't adapt fast enough genetically, okay? So that's the bottom line that I'm leading to, all right? And you can see in Sicily, Palermo, the food that is available, the fish, the fresh fruits and vegetables, and then in Japan, the fish, the fresh fruits and vegetables. And then in America and now all over the world, this calf looks at its mom and says, "Mom, is it Dad?" as it points to the Big Mac. And look at that Big Mac. The bun is without fiber, without nutrients. It's just been bleached out of all of the nutrients found in the ingredients to make bread. Then you see cheese. In America, we don't have wonderful cheese as you have in in Europe. You buy a box of Velveeta cheese, and there's 27 chemicals listed. And you ask the question, where's the cheese? All right? And that hamburger, it's grilled. And when you grill any kind of protein, whether it's a tobacco leaf in your cigar or in that Big Mac, you pyrolyze the protein to many different chemicals, including polyaromatic hydrocarbons, which is a carcinogen, okay? So when you're eating that, not only are you getting too many calories, you're getting the wrong kinds, all right? And again, I don't want to spend time to really focus in on all of these details because I want to show you visually what's really the problem here, or in my interpretation. First, as a toxicologist, I want to remind you there's only 3 ways of toxic mechanisms by which a radiation, a chemical, or a biological toxin, when it interacts with the body and our cells, it can only mutate the cell, it can kill the cell, and more recently we know it can alter the expression of genes at the translational, the transcriptional, the translational, and the post-translational mechanism level. In fact, now the microRNA is the classic example of post-translational modification of a normal gene. Let's go back to what we're really talking about when we mean epigenetics. René Debow, the famous microbiologist, Nobel Prize winner, 1969. We resemble our progenitors because we derive from them our genetic endowment. But our genes do not determine traits by which we know a person. They only govern the responses that that person takes to the environmental, and I've added, or dietary stimuli. Individuality progressively emerges from these responses. I thought that was just so beautiful before we even knew about DNA methylation and histone modification. Now, Jim Cleaver and I, back in I think it was 67. By the way, the Nobel Prize was given just last week to some other people, but we were the first to discover the human syndrome. I don't take that away from the Nobel Prize winners, by the way, but this is a fact. Jim and I noticed that these syndrome, xeroderma pigmentosum individuals, are predisposed to UV light-induced DNA damage for which they don't have a gene to repair that damage, and the cells that survive have mutations that ultimately lead to cancer. And I want to illustrate this point by saying you can inherit the gene, but if it doesn't get exposed to the environmental factor, there's no big deal. Look at this same individual, nude, and you see the only cells that manifest the disease state are the cells not exposed to the sun. And two twins that were born and diagnosed in utero as being xeroderma were told by the doctor and scientists not to let those boys ever to go out in the sunlight. So these boys have lived their lives as Dracula. They play tennis at night, they do everything at night, they have no skin cancer, but they have the defective gene. Genes and environment do determine our phenotype. Now, Bloom syndrome. This is very important for you to understand. Mutations do cause and contribute to many human diseases. However, this syndrome is predisposed to every cancer of the body, not just skin. But they have normal DNA repair. What's the problem? Their stem cells, when they have to replicate, will randomly throw in the wrong base, causing mutations. So xeroderma is a syndrome described by errors in DNA repair. Bloom syndrome is a syndrome characterized by error in DNA replication. So every time our stem cells are dividing, it can form mutations. And as I talk to you, 1/3 of you are going to get cancer before you die. 2/3 of you will die before they diagnose the cancer in you, all right? And if we all live long enough, we'd all get cancer because we all have initiated cells in our body. And the older we get, like me, 77 years old, I got plenty of initiated cells. This one, as we all know, Down syndrome, is absolutely fantastic in terms of epigenetics. They inherit 3 chromosomes with normal sets of genes. In other words, it is a chromosomal mutational inheritance. But the 3 chromosome 21 genes are normal, but they're expressed abnormally, especially during development. The baby is born hefty. They later become predisposed to diabetes, atherosclerosis, bone deformities, mental retardation, and it was mentioned earlier, the children will are predisposed to aging, premature aging, and now today, because they are living longer, a huge percentage of them are dying of Alzheimer's disease. So without having specific mutations for these classic chronic diseases, how else are you going to explain it? Those 3 sets of normal genes are out of control in terms of expression. So there is this unique syndrome, all right? Now going back to evolution again. When we became Homo sapien, we learned how to light a match, domesticate animals, grow crops, but we went out of Africa, went to France, went to Siberia, went to Detroit, Michigan, went to South America and the Amazon. Others went all the way to the deserts of North Africa and then finally to Australia. And those people who survived in those unique areas had to have energy for life. That means they had to eat the food that was found in those different areas. And you've all seen, I think it was in Nature or Science magazine 2 weeks ago, they've discovered a gene in Inuit Eskimos in Greenland that have a gene that all of us Caucasian Europeans do not have, that metabolizes the blubber that they eat, all right? But today you find Eskimos in Hawaii, all right? You find Arabs in Detroit. You're finding you having today a global diaspora of ethnic groups carrying their genes that were modified or adapted to the areas for thousands of years and now are put into another environment, cultural, which means the food is different. And I just give you examples, and finally ending with this one here. This family was given money for food they would normally eat in 1 week. And if you look carefully, you see Coca-Cola, beer, potato chips. You don't see vegetables, fish, or fruits. This is typical of the Western diet today. Now, let's quickly go over the pre-human diet, because this is important. That person who was trying to get food for life, when he found that food, he gorged himself because he didn't know whether he'd find food the next day, all right? Secondly, they only could eat what they could find in walking distance. If they had to spend 4 or 5 days walking to find food, if it didn't have enough calories or nutrients, he'd die. Here's an important thing that most people ignore. They only ate during the day, because if they went out at night, they would have been eaten, all right? But today, no one of you, and especially women today around the world, are working at night as nurses, doctors, truck drivers, police persons. And if you don't sleep at night, you don't make melatonin, a powerful natural brain hormone, which is an antioxidant. This is why some epidemiologists claim that women who work at night are at higher risk for breast cancer. And I'll come to that later. You ate seasonally available foods. I can eat blueberries in Michigan at 4, 5, 20 below zero when they're not grown in Michigan, but they're shipped in from Peru. So we're all eating foods which are now following the same diaspora all over the world. I can eat a durian fruit from Thailand. I don't even know how to eat it, but I can buy it, all right? We ate grains, nuts, fruits, so forth. And our ancestors didn't eat meat by grilling. Only today, when we learned how to make a match or light a fire, did that come into our diet. And it was basically low caloric. Today, the modern diet, it's calorically unrestricted. Foods are processed. Foods are found year-round. We eat 24 hours a day. Red meat is basically grilled or fried. And unfortunately, especially in Europe and the United States, women, to control their weight, are eating food supplements to get these so-called bioactive nutrients without the calories. And that ain't good either. I borrowed this slide from Dr. Zerhausen, the Nobel Prize winner from Austria. He was the one who linked viruses with cancer. And this, I think, demonstrates the power of nutrients and diet on human health. If you plot colon cancer, from all the countries that are producing cattle, Australia, America, Canada, I would put Argentina up there, you find the colon cancer frequency is really high. It's starting to go down in the United States because we're eating more sushi. It turns out, it's true, okay? Right after the Second World War in Japan, after the bomb was dropped, there was a lag time, but all of a sudden, when the Japanese started making, Toyotas and Sonys and so forth, they became fairly economically powerful, and notice the colon cancer frequency is going up because they're switching to the Western diet. Look at that. And years later, after the Korean War ended, the Koreans didn't have Korean barbecued beef before then. What they had was shared with the American Marines in the foxhole, which was Spam. That was the first time they got exposed to it. And now 20 or 5 million cans a week of Spam are sent to Seoul, Korea. But it turns out they're now eating grilled meat. And of course, colon cancer frequency is going up. Now here's the interesting one. India, a Hindu nation, which doesn't eat red meat, presumably, but you saw the movie Slumdog Millionaire, and you know now the rich Hindus of India are beginning to eat red meat. And look what's happening. I go back to these two pictures. It turns out before the bomb was dropped in 1945 in Japan, women who ate the traditional Japanese diet were born small. They reached puberty and didn't have breasts. And because they didn't have breast stem cells to make breast cancer. And therefore they lived longer. And because they live longer, now they have osteoporosis. And I wish I had time to tell you the link between the two. And it goes to the Barker hypothesis, is that the Japanese diet has two powerful bioactive compounds. One is genistein, the other is Bowman-Burke inhibitor, both of which are known to differentiate in utero, the female fetus's organ-specific stem cells of the breast. They differentiate in utero. So when they're born, they don't have the anlaga for breast tissue when they reach puberty. And by the same token, that same bioactive compound, or those compounds, cause the bone cells, stem cells, in utero to differentiate. That's why these young women or babies were low in stature, small in stature. Today, this typical Japanese woman who no longer eats sushi, eats mocha, drinks green tea, nor her kids are going to live long as her predecessor, who contributed to the longest median lifespan on Earth. I know I'm running out of time because I really want to get to the fundamental new experimental data. All I can say is bacteria communicated with each other via secreted factors, which then bound to a receptor on the bacterium and set intracellular signals to tell the bug to divide or do something. But when we became a multicellular organism, we inherited the genes to make stem cells and to communicate by a different sort of mechanism through the structure called a gap junction. In terms of cancer, there's two theories of cancer, and I'm not going to mention one because I think it's nonsense. It turns out we have organ-specific stem cells in every one of our organs, our eye, our brain, our breast, our prostate, everywhere. And they're constantly being damaged, like xeroderma, and if it's not repaired, they form a mutation in the stem cell. Or if they're like the other syndrome, whenever that stem cell divides, it forms a mutation. The cell doesn't care. A mutation is a mutation no matter how it's formed. Now, all of us have these initiated cells in our body, but they're usually suppressed from growing or getting into trouble because they're surrounded by and communicating with normal cells. But we now know there are many ways by that single initiated cell can escape the suppressing effects. One is chronic inflammation, one is surgery, the other is cell death. You drink too much alcohol, you're going to kill cells in the liver. You get sunburn, you're going to get too many cells in your skin dying. And babies who are born are exposed to hormones and growth factors. They need them to grow. These are also tumor-promoting chemicals. And finally, all the chemicals you and I have just eaten for breakfast and lunch and during the break, these have the ability to also block cell-cell communication, allowing this to clonally amplify into a nodule in the breast, a polyp in the colon. This is the pre-malignant lesion you and your physician notice. Finally, it becomes a cancer, and I won't go into that. But you find these gap junctions in every organ. You need them, for example, during pregnancy, all of a sudden the smooth muscle cells just are loaded with gap junctions, and the smooth muscle cells start to contract and synchronate. You have them in the heart. So when you drink too much espresso in the morning, boom, your heart starts to palpitate very fast because the caffeine in the coffee or the espresso increases cell communication. I can't spend much time on this because I got to get to the last 3 slides. It turns out we now know you can block communication with all kinds of chemicals. This is the famous, here it is, TPA. It's the croton plant oil that causes skin tumor promotion. You put it on normal cells that are communicating, as evidenced by dye transfer. The higher the concentration is, you block the communication, and that's when you have problem. There's a dose dependence, and it's reversible, by the way. And here is the specificity. It turns out these gap junctions can be affected by molecules. Every one of us, if you smoke or eat a Big Mac, you're going to be eating 1-methylanthracene. And when you put it on normal cells, which communicate beautifully, you can see they don't communicate. And anthracene, which is the most prevalent PAH in cigarette smoke, is probably the reason why we get lung cancer. And there's a whole bunch of chemicals that do just the opposite. They increase communication. And I'm going to pass this by quickly, only to get to the red wine story. All right, I'm in Italy, got to do it. Here you have the normal cells, they're communicating perfectly. You put the chemical in cigarette smoke, and you see the communication is blocked. Now you put increasing amounts of 1-methylanthracene with your resveratrol, and you see communication comes back. Now you have to be careful. All right? So if you're smoking, you drink the resveratrol or red wine at the same time, all right? And number 2, if a glass and a half is good for you, the whole bottle ain't, all right? Because alcohol blocks gap junctions. So you got to understand that. And then now this is the important, I'm going to have to miss the microbiological implications of fiber here in green tea. Get to the Barker hypothesis in the last minute and a half. About 5 years ago, Time magazine had this cover, Why Breast Cancer is Spreading Around the World. If you read the article, you'd think that young women being exposed to all these pollutants out there is causing this epidemic of breast cancer in the world. At the same time, in the journal Endocrine, it says, does breast cancer start in the womb? Wow, it's exactly opposite of this. And then my wife and I were in Buenos Aires, and I walked by a kiosk. Time magazine came out just last year with how the first 9 months shape the rest of your life. Now you women who have breast cancer can blame your mother, because this is the problem with the Barker, well, not the problem, this is the explanation the Barker hypothesis gives us. And finally, When the epidemiologists a couple years ago showed that people who were taking metformin, a natural compound to treat type 2 diabetes, for 10 years or more, it's cheap, it's non-toxic, and people were taking it religiously, they found they were at low risk for diabetic-related cancers, breast, pancreas, and liver. And I jumped on that immediately because we were the first to have human breast stem cells, human breast cancer stem cells. And it turns out, you can see when you put the individual cell in culture, in suspension, 5 days, they form mini breast cancers. If you expose them to estrogen, TCDD, or bisphenol A, boom, they grow. But now you expose them to metformin, here it is, you can see it blocks the promoting effect of the environmental promoter. And here is a more dramatic one, getting to eating at night. It turns out, I give this credit to our German and Brazilian students who are working on this. Here you have the individual breast stem cells. After 5 days, they grow into nice mammospheres. Here's the vehicle, they're the same size. And when you put melatonin, the brain tumor, the brain hormone, you can see there's virtually no effect. But now when you expose these control cells to estrogen, boom, they grow. And when you expose them to bisphenol A, boom, they grow. But when you expose them to estrogen and melatonin, that is to say, a woman is sleeping at night, those breast tumors do not grow. And if you expose the breast tumor mammospheres to melatonin and BPA, they don't grow. So I have to end. I could show you a few more slides like this, but I won't. Let me just end in conclusion by saying the quality and quantity of adult stem cells can be disrupted during in utero development. When you talk about epigenetics, you don't only talk about methylation changes or histone changes. You're going to have to talk about the numbers of stem cells which have different methylation changes than their progenitor or differentiated cells. So just by shifting, increasing or decreasing the number of stem cells in utero in any given organ, you're going to affect later in life development because either you got too many stem cells or too few stem cells in the brain, in the breast, the prostate, wherever. So that has to be taken into account. Normal human adult stem cells, which are naturally immortal, can be blocked from terminal differentiation. This is the cancer story. This disruption can lead to either an increase or decrease of risk to chronic diseases later in life. Two people can eat the same diet, smoke the same cigarette, but if you don't bring into account all these other factors, the nutrients, these anti-chemopreventive agents in the food, for example, or eating it in the daytime instead of at night, that will help explain why there seems to be a discrepancy when epidemiologists look at samples of people that seemingly have the same background. And lastly, the disruption can be influenced by the interaction of genetic factors with environmental, dietary, pharmaceutical, microbial, and lifestyle agents during pregnancy. And I end with this quote. While it has some hyperbole in it, when diet is wrong, medicine is of no use. This is why most chemotherapeutic agents don't work when you already have the cancer. When diet is correct, you don't get breast cancer, for example. Medicine is of no need. Thank you for your attention.
Thank you very much, Dr. Trosko. I think we have time for a few questions. Please wave your hands if you... Okay. Well, you talked about red wine, May I go back? Well, you talked about French paradox. It would be an issue by itself, but we have no time to deal with that. I just would like to bring some precision. Actually, resveratrol is synthesized by grapes in response to infection. And if you drink good wine, you shouldn't find much resveratrol in it. So if you need to use resveratrol from wine, you could expect to have to drink between 50 liters to 100 liters a day to get some effect, but in doing so, you may shift the risk-benefit balance of resveratrol.
Right. True. In the United States, I might add, you can buy capsules now of red wine that are equivalent to 100 bottles of red wine, all right? But here's another problem we all know philosophically that imbibes our culture, and that is if one pill is good for you, 10 must be better. And all of a sudden, those antioxidants that are supposedly in this pill now become pro-oxidants. So there's no easy way to deal with this. If there's no other question, I just have to end on the quote from... Oh, you do.
Thank you, Dr. Trosko. Nico van Belsen, IDF. You spoke about not consuming grilled meats. I assume you know about this hypothesis that the human brain could only grow to its size when we discovered how to process food, how to cook, basically, because that liberated more nutrients from our diet. So could you kindly comment on that?
Well, that's a real nice philosophical question that you shouldn't have asked me. This will go on for an hour. Remember, those people who learned how to make fire needed that food, and they didn't live very long. So they didn't live long enough to get the cancer, but benefited from the short-term calories and other things, okay? That's a short answer to that.
May I have a follow-up question, please? Yeah, just a follow-up. I must admit, I don't know precisely how long it is that people invented fire and cooking, but it must be, I think, at least 30,000, 50,000 years ago.
Right.
To give an example, about 10,000 years ago, humans invented collection of milk from animals and drinking that milk.
Right.
Within that 10,000 years, 1/3 of the human population became lactase persistent, which is the opposite of lactose intolerance. This shows how quickly the genome can change. Second, the benefits of milk, but that's a different story. But coming to this point, how quickly the genome can change. Suppose there would be bad effects of cooking, we would have had 30, 50,000 years to kind of adapt ourselves to that, right?
Well, remember, when you have a population changing their diet like that, you see a change. In that population, you may have just one individual who has a mutant that allows him to survive where the others died. The bubonic plague is a good example of that. Those that survive then leave offspring that have that trait, all right? So that could have, mutations in a given gene don't happen throughout the population very fast. It usually happens in one person, all right? And that person has to survive to reproduce, all right? But the other explanation, it may be hereditary epigenetic changes too. We can't rule that out right now, or I can't.
We have two examples. It's lactase in the gut and amylase in the saliva. It takes about a few thousand years to modify. Has anyone any further question? Looking carefully? No. So thank you again very much, Dr. Trosko.